Hello again. For those of you that have just joined us, my name is Tirth Patel, Vice President of Investor Relations, and I will be moderating today's discussion. Welcome to Quantum Genomics webcast and conference call to discuss the Phase 2A clinical data in hypertension of their lead compound QGC001, the first orally active aminopeptidase A inhibitor. Quantum Genomics is a leading biopharmaceutical company with the mission of developing new therapies for unmet medical needs in the cardiovascular space. The Phase 2A data being discussed today was first presented yesterday by the principal investigator, Professor Michel Azizi, at the 2017 European Society of Hypertension Conference in Milan, Italy. Management will do a question and answer session immediately following the presentation. All questions can be submitted online on the webcast platform. For listeners dialed in, please refer to the webcast link found in Quantum Genomics' press release to submit a question. Questions may be submitted at any time from now on and throughout the presentation of results, but please keep in mind they won't be answered until after the presentation. Before we begin, I'd like to remind everyone that management's remarks today may contain forward-looking statements. These include statements regarding the company's clinical development plan for QGC001. Such statements are based on management's current expectations and are subject to a number of uncertainties and risks that could cause actual results to differ from those described in these forward-looking statements. Factors that may cause such a difference include, but are not limited to, those described in the 2016 annual report of the company. Now, I'd like to please introduce Mr. Lionel Segar, President and CEO of Quantum Genomics, and Dr. Bruno Bess, Chief Medical Officer. Lionel?
Thank you, Tirth. Good afternoon, and thank you for joining us for QGC001 Phase 2A results presentation. Today, we are one step closer to fulfilling our mission of developing a potential therapy for the treatment of high blood pressure. We have reached a new milestone. Our agenda for today's call will include a reminder of the mechanism of action of the new class of drug, the effect on animals, and of course, the analysis of our QGC001 Phase 2A study in patients with hypertension. We will also outline the impact of these results on our plans for advancing the development of QGC001 in hypertension patients as well as heart failure. QGC001 is the first drug candidate in a new class of drugs, brain aminopeptidase A inhibitor. So it's slide 3. Is that okay, Tirth? I don't have a feedback on the slide. Slide 3. QGC001 is the first drug candidate in a new class of drugs, brain aminopeptidase A, so-called BAPIs. BAPIs are innovative drugs that target the new central pharmacological pathway leading to both anti-hypertensive effect and cardioprotection. As a reminder, BAPI achieve a triple mechanism of action with one single drug. Blood pressure control involves multiple regulations. To positively impact this regulation, BAPIs prevent the conversion of angiotensin II into angiotensin III at brain level, which results in decrease in the vasopressin release, decrease in sympathetic nerve activity, and the regulation of the barrel effects, acting on diuresis, vascular resistance, and heart rate. On the left-hand side of this slide, you can see the results in animals. We have demonstrated that there is a decrease in blood pressure in 2 different animal models, SFR rats and docasophiles, and no change in blood pressure in normotensive rats. The effect on blood pressure is more important in docasophiles, which is a mimetic of the salt-dependent hypertensive patients. In human, we did 124 healthy volunteers in several different phase 1 trials. We've seen no change in blood pressure in phase 1 in non-potential volunteer in heart rate or renal function. Next slide. We believe that one of the key components of treating hypertensive patients successfully is to target a specific profile, patients with low renin, high vasopressin, or LRHV. Our goal is to launch a phase 2 trial targeting these LRHV patients where the unmet medical need remains huge. We are committed to making a difference for these patients.
Hello, everybody. We have fixed our technical difficulties, and on the line we have Mr. Lionel Sigard and Bruno Bess back online. Thank you.
So we are on the slide 5. The aim of this pilot study in hypertension with the GC01 was to confirm a proof of concept obtained in animals, also to identify predictors for blood pressure response in order to design further trials. The study limitation was the small sample size. It was a pilot study, but based on power calculation, and I will let Dr. Bruno Bess comment on this later. Of course, a short duration of treatment, 28 days. And the idea was to have a fast validation of proof of concept in human, an unselected population, which is also a study limitation, as we plan to target low renin high vasopressin patients with our drug. Leave the floor to Dr. Bruno Bess.
So hello, everybody. I'm on slide 6. The study was a pilot, randomized, double-blind, placebo-controlled crossover study. All the patients stopped their medication 2 weeks prior to the inclusion into the trial and enter a 2 weeks placebo running period, and were treated after with 250 milligrams BID of UGC001 for 1 week. Then the dose was increased to 500 milligrams BID for 3 weeks, or a matching placebo. The primary endpoint of the study was the difference between placebo and UGC001 in change from baseline to day 28 in daytime ambulatory blood pressure. And 4 centers participated to the study in France. Patients who were selected were male or female between 18 and 75 years old, suffering from mild to moderate hypertension, stage 1 or 2 hypertension, with a normal renal function. On the next slide, slide 7, you can see that 40 patients were screened, 6 were not included, so at the end, 34 patients were randomized, and this is the intention to treat and safety population. 4 patients discontinued during the course of the trial, 2 for adverse events, 1 withdrawn consent, and 1 for very high blood pressure over 170. And 1 patient had a protocol deviation, meaning that 29 patients can be analyzed protocol analysis. As you can see on the right side of the slide, mainly male Caucasian patients were recruited. The average age was 57 years, 2/3 of male, and 90% of the patients were white ethnicity. The baseline of his blood pressure was 148 millimeters and diastolic 93. It means that we finally selected patients with a very mild hypertension. On the next slide, which is slide 8, you can see here that at baseline, the blood pressure was similar in those groups, placebo and treated patients. The blue line with placebo is similar to the red one. The next slide, which is slide 9, is probably the most interesting slide about the results. On the right side, you can see the results from the multivariate analysis on the change from baseline on ambulatory blood pressure. And as you can see, the baseline blood pressure had a significant impact on the change, the decrease in blood pressure, as well as the QGC-001 treatment with the p-value of 0.06, whereas there were no impact on the initial level of active renin, meaning that whatever the level of renin, the results are the same, and no impact of age, gender, ethnicity. Remember that we have 90% of white people, renal function, or other hormone. On the left side, very interesting, as you can see, the red line showing that the higher is the initial blood pressure, the larger is the decrease at day 28. This is the case in red in the treated group patients, but it's less obvious in the placebo group. And you can see that the difference between placebo and active increase with the level of QGC-001. And this is really consistent with what we found in animals, because in hypertensive animals, the aminopeptidase A is overactivated in hypertension. On the next slide, which should be the slide 10, you can see the two graphs at day 28 of the ambulatory blood pressure. And as you can see here, the red dots, which represent patient treated with QGC-001, is always below the blue one, which is the placebo, whatever the time. And the primary endpoint, which is the average daytime ambulatory blood pressure, shows you the difference between placebo and active of 2.7 millimeters. And for the office blood pressure, not shown on the graph, the difference is 4.7 millimeters. With a p-value around 4.15 for both. Let me stop a little bit and comment on the p-value. We usually do not display a p-value for a pilot study, and particularly, which is the case here when the sample size was not formally calculated based on power calculation. But what does it mean? It means that we have 16% of chance that the difference we observed between groups is related is obtained by chance. And if you remember the previous slide, when the treatment was adjusted on the baseline blood pressure, the p-value is now 0.06, meaning that only the difference may be due to by chance only by 6%. On the next slide, slide 11, just reminds you that we observed no change in plasma hormone, active renin, aldosterone, cortisol, ACTH, peptine, or apelin, either in QGC-001 nor in placebo. And this is really interesting because it confirms that the original mode of action of QGC-001 is not mediated by renin or by aldosterone. On the next slide, slide 12, you can see the comparison in safety profile between QGC-001 and placebo. To summarize, the safety profile of GC001 was similar to what was observed in the different phase 1. Only 3 serious adverse events in GC001 as compared to 2 in the placebo group. And interestingly, only 2 rash, only in 1 in treated group, 1 in placebo. Another very important point is the lab test and the lab test tolerance. As you can see on the slide, there is no decrease in potassium nor in sodium, which is very interesting in not only in hypertension but also in heart failure and especially the sodium. And there is no change in renal function, even a trend to improve creatinine. Let me move on to the slide 13. This slide illustrates the consistency between what we obtain in experimental trials in animals and in humans. On the left side, what we obtain in rats. As you can see, there was no decrease in blood pressure in normal rats, whereas we observed a decrease in SHR rats, and a more important decrease in DOCA salt rats. Just to explain that SHR rat is a hypertensive model, animal model, which is a standard hypertension, all-comer hypertension, whereas DOCA salt is more close to the target that we want to study with GC001. And in humans, we found exactly the same. In healthy volunteers, with normal blood pressure, no decrease in blood pressure, whereas in this unselective population study here, there is a decrease in blood pressure. And the next step for us is to study a more selective population, so-called low-renin high vasopressin, closer to the DOCA salt population in rats. And this is what we will study in our new herb study that will start in the US in autumn. It's a longer treatment. Yes. On slide 14, Lionel already say a few words about study limitations. As said, this was a very small sample size, only 34 patients, usual for a pilot study. And we are in the field of hypertension, where there is a high inter- and intra-subject variability. So finally, there is a very low power for this study, but once again, the sample size has not been formally determined based on the power calculation, and we did not expect to have a statistically significant effect. If we would have done that, of course, we should have needed several hundred patients in the trial. This is a very short study. Treatment duration is only 4 weeks. And what we observed in the study is that the effect is larger at day 28 as compared to day 14. And we may expect that with a longer study, the decrease in blood pressure would have been higher. And usually in a hypertension study, treatment duration is 2 to 3 months. But we wanted to have a fast validation of the proof of concept in humans, confirm what we observed in animals. That's why we decided to go fast with a small study and short duration of treatment. And moreover, as we discussed before, this was a non-selective hypertensive patient, not specifically a low-renin high vasopressin, and which is closer to the SHR model in rats. And the baseline blood pressure was quite low. Remember, the average in blood pressure was 148 millimeters, and the effect of QGC-001 increased with the level of baseline blood pressure. So that may suggest that for patients with higher blood pressure, the effect would have been higher. So in conclusion, on slide 15, in the multivariate analysis of the data, the two main predictors of blood pressure decrease were baseline blood pressure with a p-value of 0.001 and treatment by QGC-001 with a p-value of 0.06, and as compared to placebo. 4-week treatment with QGC-001 decreased daytime ambulatory blood pressure by 2.7 millimeters, decrease office blood pressure by 4.7 millimeters, had no effect on hormone involved in cardiovascular and especially hypertension regulation, and was safe. So finally, the last slide is slide 16. Grade aminopeptidase A inhibition elicits an anti-hypertensive response in hypertensive patients, as we already demonstrated in animals, and it is very important that opens the way for this potential new class of drug to treat cardiovascular disease, including hypertension and congestive heart failure. And we are really confident in this result and very happy to get a confirmation of what we observed in animals. That's why we are ready to launch a larger phase 2 trial in the US based on FDA advice that will include 250 hypertensive patients, and this time with a higher cardiovascular risk. And the design of this study will be released by the end of the month on June 27th during our KOL Day event. Thank you very much.
Time for question.
All right, thank you all. Now we will host our question and answer session. Thank you, Lionel and Dr. Bruno Beth. Our first question comes from Jason McCarthy. How many patients on slide 9 had higher blood pressure at the baseline, 180 or higher, where you saw the greatest effect of QGC-001? And he also asks the second question, for the New Hope study, will the treatment duration be extended to 2 to 3 months? If so, what delta in blood pressure would you target?
Okay, so for the first question, if you can perhaps go back to the inclusion criteria, or I can just comment without the slide. The patient, in the inclusion criteria, we want to include stage 1 or stage 2 hypertension patients. So it means that the blood pressure should be below 180. So the answer, how many patients above 180, the answer is only one, exactly 180. And the second, but if we really want to know what would be the effect in patients with higher blood pressure, of course, it's difficult to present subgroup and a non-planned subgroup analysis. It's not really meaningful. But if we just take the threshold of 148 millimeters, which is the median, meaning that half of the patients were below at baseline, half were above. In this case, for the patients above 148 millimeters, decrease in blood pressure, in ambulatory blood pressure, with QGC-001 is minus 5.3, and in this subgroup of population, half of the population, the p-value is 0.01. And interestingly, we got similar results for the diastolic blood pressure with a decrease of 4.3 millimeters with the QGC, the p-value of 0.02. And the second question was about the NEWOP study. So the treatment in the NEWOP study will be 2 months. We will present the detailed design of the study at the POL day at the end of the month.
Great, thank you. We also have another question. You said...
Sorry, go ahead.
You said that the higher the initial blood pressure is, the larger the decrease. Can you give us an example of the decrease when patients are more hypertensive?
Yeah, this is exactly what I just said. If we select only patients with above median baseline blood pressure, so 148, the decrease is minus 5.3 with the treatment.
Okay, thank you. Next question comes from Mr. Bertrand Delsu. First question is, what was the grade of the SAEs and were they considered treatment related? Were they considered treatment related?
So the grade was mild, and both events, either in placebo or in QGC group, were considered as possibly related to the treatment.
Okay. Question 2, also from Bertrand Delsu. Was there effect on diastolic blood pressure?
The effect on diastolic ambulatory blood pressure in the overall population was very small, but in the patient with the highest initial blood pressure above median, so half of the patients, the decrease in diastolic blood pressure was minus 4.3, the p-value of 0.02, sorry.
Great, thank you. Question 3, are the data and significance calculations computed on a per protocol basis or on an intent to treat basis?
So all what we presented here was based on the ITT population. But of course, we have done the same analysis on the per protocol populations, and there is a very small difference, but which is very much expected because there is only 5 patient difference in the group, ITT and per population and per protocol. But of course, in per protocol populations, the results are slightly better.
Thank you. Question 4. Is the reason on a lesser effect on nighttime related to a lower nighttime baseline blood pressure?
So of course, we do not know the exact reason, but what we may expect is that because always the blood pressure is lower during nighttime, the effect of QGC-001 is lower because, as we said, the higher the blood pressure, the larger the effect.
Thank you. The next question, should you be considering a dose increase in the next phase 2? What was the rationale for 500 milligrams?
We will have as well a dose increase in the next study. But of course, we will talk more about the study design later. And the rationale for the dose was based on the animal models and the doses in animals. So this is the equivalent dose from the animal models.
Thank you. And the last question from Bertrand is, do you think the washout had an effect on the baseline after the first part of the study?
Yes, of course, because the patients were treated. So when the treatment was stopped, the blood pressure increased. This is the reason why we want to have a washout period.
Thank you.
Or perhaps, if the question was the washout period between, because not the initial washout period, but the period between the two groups, there is no period effect for the crossover design, no statistically significant period effect for the crossover design. So the answer is no in this case.
Great, thank you. The next set of questions comes from Max Jacobs, Quantum Genomics covering analyst at Edison Investment Research. Max asks, were patients on background antihypertensive medications? If so, what were they, and were there any limitations on that background medication?
So all the medications, whatever the treatment they receive, all the medication, as I said, was stopped 2 weeks prior the enrollment into the study. During the course of the study, the patient only received CDC-001 or matching placebo. It's not on top of.
Okay, thank you. His other question asks, what level of blood pressure lowering will you need to achieve clinical significance?
With the same sample size? Oh, yeah, you don't have because the p-value level is linked to two different things. The first one is the difference you may expect, and the second one is the sample size. So, to get a significant, it's impossible to answer, but 34. Okay, let me say. If we want to have with the same difference to show a significant difference, as I said, we would have need several hundred patients. And with 34 patients, to get a statistically significant, the difference between groups, I did not calculate it, but it should have been huge.
Thank you. The next question comes from Mr. Jean-Christophe Lapivert. What is the percentage of low renin, high vasopressin patients in this study?
So I would be very happy if I could answer this question. But to be more serious, what are the patients with the low renin, high vasopressin? What we know from the literature is that this is more likely in elderly, and this is also more common in minorities, including Afro-Americans, Hispanic, Asiatic patients. So here, all the patients were relatively young, below 75, no true elderly patients, and most than 90% white patients. So very few patients were probably low renin, high vasopressin.
Thank you. Got one more question from Mr. Christophe Dambou. Can you please give us more information about the European Phase 2 study?
In our failure, okay. So the study is running in Europe. The study is currently running, and we expect to have the result by the end of next year. We are not disclosing it. Yes, it's in 2018. We are not disclosing any information on running the study. Only 75 patients will be included in 8 European countries.
Thank you. One more question from Christopher Dombu. Which parameters were taken into account in the multivariate analysis?
So this is a very easy question because on slide 9, you see all the parameters that are included in the multivariate analysis. So I can quote them: baseline blood pressure, treatment effect, baseline renin, age, gender, ethnicity, renal function, copeptin, and apelin. And to answer in a more correct statistical way, the model presented here is the full model, and we underwent a backward procedure to select the significant parameters. And so the two only significant are baseline, daytime ambulatory blood pressure, and GC.
Thank you. Our last question comes from Mr. Siwit Chen. Is there any criteria for the p-value? Less than 0.05, it seems to me that 0.16 is relatively high.
So I can comment again on this point. So 0.16 means that we have 16% of chance that the result, we have 60% that the result may be observed by chance. With that. So the difference is due by chance. So this is relatively high, but this is usual in a small study with 34 patients. Because to get a significant p-value below 5%, which is the usual value, we would have need 200 or 300 patients in the trial, and we want to conduct a small, fast pilot study to validate the proof of concept and not to get a significant p-value. And in the multivariate analysis, once we adjust the effect on the baseline value, the p-value for the treatment is 0.06, which is not far from 0.05.
Thank you. That concludes our question and answer session for today. Thank you all for joining, and I'd like to remind everyone that Quantum Genomics will be hosting on June 27th in New York City an Investor and KOL Day event where management and KOLs, key opinion leaders in the industry, will be discussing this data in further detail and an overview of the next clinical trial in hypertension and how it is being planned. So if you would like to attend, please send myself, Tirth Patel, an email or a message immediately after this presentation, and we look forward to your attendance. The audio recording of today's presentation will be available immediately following. Thank you all for listening. Goodbye.